Start over. Hi, we're live. Thank you, Janine, from the alumni office for recording this. Coordinating this, we have some folks that we're streaming to on Zoom, possibly even Dr. Veith. I'm not sure because I can't see. Okay, yeah. And he's been a generous donor to behavioral neuroscience. In part, he has a lot of things on campus, some other activities as well in his family. And part of it is supporting our ability to have different seminar speakers come in from kind of the PNW local area. So I'll get started here. I'm really happy to have Dr. Melvin Rouse as our speaker. I've known Melvin for a few years. I'm not a bird guy, but I'm a rat guy. But Melvin and I have served on a committee in the American Psychological Association together, where we go in and try to inform lawmakers, which is really hard, about the importance of biomedical research. So we've been to Capitol Hill a couple times together in Washington, D.C., and it's been all good times. But unfortunately, things are complicated. That's not the focus of the lecture today. Dr. Rouse is in the Department of Psychology at the University of Puget Sound in Tacoma, beautiful campus if you haven't been down there. In terms of his education, got his bachelor's from Virginia Tech in psychology, his master's from Boston University in 2005, also in psychology, then a PhD at Johns Hopkins University in 2014 in psychological and brain sciences, a little blurb, and he knows I stole this from his website. Dr. Rouse's research looks at how hormones, the brain, and reproductive behavior interact. His lab uses Songbirds as a model system. This model is unique in that it allows for the ability to study how gonadal hormones act to modulate patterns of learning and behavior, as well as how hormones affect the perception of behavior. These studies demonstrate the influence of the endocrine system on brain plasticity, learning, and social behavior. We have the title of the talk here, Hormones and Sex, the role of testosterone in the instantiation of singing in female songbirds. Please welcome Dr. Rouse.
All right. Hello. You can say it back. Hi. Okay, one more time. Hi. Hi. Okay. See, I always start my classes like this because I like to get the students going, and it's like it's a give and take, back and forth. Thank you for introducing me, Jeff. I always forget what's actually on my website. So I was looking at the bio. I was like, oh, I actually updated that. I didn't get through to the folks on technology services. But yes, I'm here to talk about hormones and birds. And I'm going to start off by saying that I'm going to do some unorthodox things in the talk because that's kind of what I do. I am a bit of an unorthodox scientist. But I want to state something that is very obvious. So I'm going to state the obvious, and that is that hormones are clearly linked to reproduction. Show of hands. Who agrees with this statement? Okay. Who agrees that it's also patently obvious? Hormones are linked to reproduction. Okay. Then why then am I asking this question that is patently obvious, you all agree with, and that there's centuries worth of literature to say that yes, hormones and reproductions are tied together? Well, I'm doing this because I argue that we have misinterpreted exactly what that means. Now, misinterpretation. What is a misinterpretation? To break that down, I am actually going to start backwards. So usually when you give a talk, you do this slow build up where you give big general information, then you funnel on down, funnel down to your specific question. Then you break down, how did you address this question? How did you answer it? What's the data? I'm going to go backwards. I am going to start with the ending, and I am going to go straight to this notion of hormone myths and hormone facts. That's going to be the first part of this talk. Then we're going to talk a little bit about sex hormones, the brain, and bird song. And then the second part of the talk is going to deal with the application of some of these things. So thinking about how do we actually think about what a misinterpretation of data is, how do we then apply this, and then what is the impact of that misinterpretation? So we're going to talk about the limits of hormones, and I'm going to talk about this point here, that there are no male or female hormones. This should not be a radical statement, but given where we are currently, that does feel a little bit radical to say. But just to kind of give the bag away, because like I said, I'm starting at the end. All biological creatures that have organs, glands that produce hormones, that secretes, in particular, steroid hormones. All steroid hormones are cleaved from one single molecule, that is cholesterol. And that means that if an enzyme is present to cleave that cholesterol into a hormone through a steroidogenesis, then that means that any steroid hormone can be made from cholesterol, meaning that any gland that produces a steroid hormone, technically, if the factors are there, can produce any and all hormones, steroid hormones, I should say. So this is going to be the basic structure of the talk. And what I mean by beginning with the ending, addressing hormone myths and facts, is right here. So there are questions I have with regards to how we talk about hormones and how students and folks who are in the lay public, how they understand hormones and what this actually means. Because when we think about biological discoveries, thinking in particular, going all the way back to Berthold and thinking about this factor that makes chickens into roosters. So you have this capon, this male rooster that has its testes removed, and then suddenly you don't get the classic male typical features. You replace that either with the hormone that was originally taken out, or the testes rather, that was originally taken out, or from a donor, and then you suddenly get this reinstantiation of the male behaviors. But since those experiments all that time ago, there's been this social impact of what that actually means. Because when we think about testosterone, if I say testosterone means male, does that sound wrong? Testosterone means male. Okay. Now I've got some folks who are astute and is like, I know where this talk is going. I'm going to shake my head now. Yes. What about estradiol? Because again, this is another bit of problematic misinterpretation of what data are. Estrogen is actually a class of hormones. It is not an individual molecule. It's a class. So if we say estradiol or one of the estrogens, female, yes? Oh, some of you have done your homework. Some of you may have read Randy Nelson's book and decided, oh, don't estrogens play a role in the masculinization of male animals in the perinatal period? Yes, that is the case. But still, we think about the lay public and the lay public's understanding of this, the way in which we talk and teach about hormones. There's this idea that there are three things that are happening. One, that sex differences, whether it be in hormone composition, the structure of bodies, that if there's a sex difference, then that means that they are dimorphic. My basic Bio 101 folks, when we say a biological dimorphism, a dimorphism means what inherently? We have two distributions, distribution A, distribution B. If it's dimorphic, these two distributions don't have any overlap. You have A and you have B, you clear categories. If they're different, even if they are statistically significantly different and not dimorphic, A and B can be right here, right on top of each other. So when we think about this, there are a few myths that the lay public has, and then that impacts public policy and how folks think about these sorts of things. But sex differences are always dimorphic. That's one myth. The second myth, that there are male and female hormones. And the third myth, that testosterone, the more testosterone, the more masculine. This last one in particular, the more testosterone, the more masculine. Does that sound right? As a bald American, I can say I have heard this before, because male pattern baldness has to do with what? 5-alpha reductase. What is 5-alpha reductase doing? It is converting testosterone into... That's all right. That's not in the quiz. It's not on the quiz. But uh, but again, thinking about this last statement, that the more testosterone, the more masculine. Because again, what this talk is trying to set up, and why I'm starting here with the ending, is in particular because how most folks in the lay public and students as well, how we think about what sex is in terms of biology, in terms of how bodies develop. We have this very traditional perspective where we have males in blue and females in pink, where we look at maleness. Maleness has to do with aggression, courtship, signaling, some notion of parental care, but really in terms of parental care and investment, it differs by animal species, and more so, how does one become a parent? Because we tend to tie maleness and male behavior not necessarily to actual parenting, but rather to the production of children. And again, in thinking about femaleness, this traditional model, we're thinking about mate choice. Who is the female going to choose to mate with? And sort of simplifying female behaviors around this reproductive idea. And so when we're looking at circulating testosterone, and in particular for seasonal breeders, animals that breed across different time points of the year, we see that in terms of circulating T, there's a measurable difference at certain time points in the year for males and females. That's true, yes. But really, when we think about what's happening with bodies, and in particular, the place of hormones, the place of sexual reproduction, sexual differentiation, it is much more complicated, where you have multiple hormones, multiple signals that are changing at different time points in the year. And that's not just true of females, this is also true of males. And all bodies are responding to different selection pressures, different features of the ecology, different mating strategies that are being employed, different parenting strategies. I work with songbirds. Songbirds have what we call obligate biparental care. If you have a zebra finch pair that where only one parent is doing the job, that couple kind of sucks. And I hate to anthropomorphize in that way, but in many animal systems, in particular songbirds, both parents are doing the work of getting progeny up into adulthood. And so when we think about different mating systems, different parenting strategies, when we look across the tropics and southern species, we start to see that in terms of how we understand behavior and how behavior is linked to brain and hormone differences, we actually might want to move away from this notion of sex roles and actually focus on what the animal is doing. What is the animal doing in its particular context and environment? And then how might the physiology facilitate those things? Rather than looking at, okay, we see this on the average. On the average, the males have this level of hormone, the females have this level of hormone. That means that this hormone that's greater in this male is going to be responsible for these male behaviors. Rather than looking at the full context and thinking critically about, okay, what might be happening here? And again, my thinking on this kind of goes back to a study, a paper I wrote with an undergrad student by the name of Evan Hamilton. Evan was a summer research student that came on board as a part of his thesis for neuroscience. He was a neuroscience major at the time. And we were thinking critically about what he in particular was taught about sex, biology, hormones, reproduction, and then how that impacted him as a student. And so we worked together to sort of think about, from a pedagogical perspective, how might we actually envision a world where we can focus on context rather than outcome? So not just looking at what happens on the average, but thinking about what is facilitating these patterns that we see across different contexts. And so that kind of takes me here, where we jump back to this point that, again, we know that hormones are clearly linked to reproduction. You all are on board with that. That is a clear idea. And in terms of thinking about what that means, when we think about what hormones are dictating different kinds of behavior, whether we're talking about parenting behavior, whether we're talking about mating behavior, different mating strategies, whether we're talking about the interaction of environmental cues and competition. So if you're trying to secure a mate and you're competing with others, how might hormones be involved in that pattern of competition? For me, that's a critical question, because when we're thinking about it, again, with the classical framework, we're taught that, okay, when we think about the machinery of reproduction, with what regulates and controls hormones, this is going to be done through feedback loops, right? So, And again, this is the tricky part about starting your presentation and working backwards. So now I'm moving into some of that more general territory that usually would precede everything. And so in thinking about that here, we have our brain. Our brain has factors like gonadotropin releasing hormone, GnRH. GnRH is within particular spots of the brain, like the hypothalamus. And then you see signaling from GnRH neurons into the pituitary, where the pituitary releases luteinizing hormone and follicle stimulating hormone into the bloodstream. Those factors then feed onto the gonads. The gonads then release hormones, and through feedback mechanisms, in this case, we're talking about a negative feedback loop, the body is then able to maintain a stable release of certain factors. Negative feedback loops are good for when we think about corticosterone or cortisol, testosterone. Generally speaking, you can see some notion of negative feedback for estradiol regulation, but because estrogens are tricky, you also have positive feedback loops, in particular for estrogens and progestins. And so progesterone in particular, when we think about progesterone and estradiol, you now have to complicate that a little bit further. But long story short, there's a system at play, right? And this system is reliant on specific factors, specific molecules. And the animal system that I work with is no different. I work with songbirds, yes? And in particular, I work with them in the context of their song, when they're singing. And when we think about birds and singing, it is about reproduction. So in particular, when I first started on canaries, and canaries have this, males have this special phrase that they, I hate the name of this phrase, but they term this bit of their animal communication, they call it the sexy syllable. I know, I know. It was a group out in France. So Michel Vallet and Eric Kreutzer, they came up with this name. But the whole point was that, okay, because this is so closely linked to reproduction, this is clearly tied to some signal that is attractive to the female. When we then look in the brain and we see the regions of the brain that control singing, and again, they were thinking about this in the male context, each of these regions that are important for this production are sensitive to steroids. In particular, you see the androgen receptor and you see both estrogen receptors. So you see estrogen receptor alpha and estrogen receptor beta. So in terms of thinking about songbirds and song, songbirds were no different than all other species. There was this thinking that, okay, we see testosterone regulating this specific aspect of male song. If you deprive these regions of testosterone, so let's say you castrate a male, now you've changed, you know, the hormone profile, and you've changed their brain, and they can no longer do this behavior. So that must mean that testosterone is regulating this specific function in males. That makes sense from a logical perspective. But again, my job here is to problematize this just a little bit further, to show you a little bit of the evidence going down that path. This is some behavior that I analyzed with some colleagues. So on this project, I was the behavior guy. I was the guy who did all the bioacoustic analysis and actually isolated the individual notes within each song to identify what was actually being sung and when. And so in this study, we're looking at male and female, well, male canaries in particular. So we only recorded the male canary song. We didn't really record what the females were doing, but the males were either paired with a female, they were alone, or they were on short day lengths and then photostimulated. So on the photostimulated side, the SDs, with these birds that are seasonal breeders, if you hold them on short days long enough, they go into what's called photorefractory, where they're now no longer in breeding condition. They're not ready to mate anymore because it's short days. It's winter. I don't want to mate in the winter. I'm a bird. What's available for me? Like, there's not a lot of food. If you're in Washington State, it's rainy and then it's cold and then it's sunny. That's great, but then it's rainy again, then it's cold, and you cycle through that every other hour. So it's not the best time to probably mate. And so when you stimulate them, you're indicating that the day length is starting to get longer. And so that length change in day length also corresponds with changes in their hormonal profile. So stimulating them with the day length triggers growth in the testes. That triggers release of testosterone. So there's an increase in the testosterone profile. And that also leads to changes in their song output. So with these birds, what I looked at in particular were the kinds of notes that they were producing. So birds that were paired with a mate, they did not really sing sexy syllables. This was the best sexy syllable you could get. And you would think like, why would that be the case? Why would the male who's paired with a mate, why would he not sing sexy syllables? Because he already got a mate. And now they're moving on to other things. So we think about the function of song in terms of a mated pair. Song then changes where we're no longer advertising to one another. In the case of zebra finches, they'll do what's called an undirected song. An undirected song is messy. It's not nearly as clean. It's not what the male uses to advertise to get a new mate. But it's important to the female to hear because it actually stimulates that pair bond. So males that engage in more undirected singing with the partner actually show more stable pair bonds than males who don't, even though the song is messy. And so canaries don't do undirected song, but you can look at the quality of the sexy syllables, and that can give you some indication that, you know, they're just like, oh, little love tap here. I'm showing some social affiliation to strengthen our pair bond. But the male who's alone, who's by himself, who's trying to find a mate, this is a very sexy syllable. So these canaries, they have what's called a biphonated syrinx. So it's split in two. To make this syllable, one side goes, huh? The other side goes, huh? Up high like that. And so they kind of switch back and forth like that really rapidly. And when I say rapid, I mean sub-millisecond difference between the instantiation of one note versus the other. And so this rapid switching back and forth, that's an indication of, oh, this male he's fit. He is good to reproduce, good to breed, good genes, and that's indicative of a life history of, resources and whatnot. So you can actually tie the quality of the song to a life history where, we're looking at resource allocation, social histories, and environmental condition where these males are healthier and they have more stuff. The males who are photostimulated, because they're in a stimulated condition, they're just now getting the machinery warmed up. So when we think about what's happening, so interestingly with songbirds, they actually have to, in this case for this species, I should say, they relearn songs across seasons. And so a new season is going to have a new song. And so this male is in the process of putting together a new composition, and he hasn't quite figured out how we're going to get these sexy syllables going, because I would like to get a mate, but I'm not quite there yet. I'm practicing, I'm warming up. And so when you look at this, you see specific kinds of changes. And I know I'm driving this home pretty hard here, but when we think about these sex differences, again, all of this stuff in males is driven by the presence or absence of testosterone. So once you put them in reproductive condition and testosterone goes really, really high, then you start seeing behaviors like this where they're actively trying to attract a female. So here's a male here. He's singing, and how you can tell he's singing is I'm a fan of Prince, the musician. I don't know if I have aged myself out where students no longer know who Prince is, but Prince was what we would call a very sexy man. And what this bird is doing, this is very attractive to this female right here. And so you can see he's inflated this throat patch. He's singing these sexy syllables. And if you were to hang around long enough, this female might do what's called a copulation solicitation display, where, again, this is a voluntary choice. This is something that the female is saying, I am receptive to sexual activity. So she lowers herself, arches her back, vibrates her wings rapidly. And if you were to zoom in on the cloaca, the feathers around the cloaca would be pulsing because, again, birds don't have intromittent organs. So you have to get two vents to line up together. And so when those cloacal feathers are pulsing, it's essentially saying, this is where you're supposed to land. Yeah. And that is a voluntary behavior. So it's a little different than what with rats, where if you have female rats in breeding condition and you engage lordosis, that's more of a reflex. So if you touch that patch on the flank, then they're going to sort of automatically lordose. This is a choice that the female bird is making based on what she is hearing. And that's an important thing to point out, because what's interesting though is that for canaries, in particular female canaries, their brains aren't different than the males. They have the same brain areas that regulate singing. And so one of the things that we did back in the lab uh, was give them testosterone. So we would give female birds testosterone, and we give them it on the range of what a male would see. So what's the average male circulating level of T during the breeding season? We'd mimic it that through a hormone implant. And what we'd see is within a week of treatment, you'd get this nice bump in singing. So on the y-axis here, we have the proportion of time spent singing. And just thinking about it practically, if you're spending roughly like 15% of your day doing one activity, that's a lot of time dedicated to one activity. So think about how long, so you got 1 hour, 15 minutes is 1 hour, let's say 100 minutes, because I can't do math, because I'm trying to think on the fly. So you got 100 minutes, and now you're going to spend 15 minutes doing one thing. That's a lot of time. And so we recorded at multiple time points during the morning, during the evening, during the midday, and during the evening chorus. And at each time, when we looked at the proportion of time spent singing, within 1 week, they're bumping it up to a roughly like 8% of their time spent singing. Females who are untreated, who are treated with blank silastic capsules, they actually do sing. And this is a controversial point a little bit in the bird community. Female birds actually sing at a more higher rate than we talk about in textbooks. So when we talk about the framing in textbooks, usually we talk about male song. Males are singing to defend the territory or to attract a mate, but females also sing in many species. Folks like Karen Odom, Katharina Rebel, Naomi Langmore, they're doing this work looking at female behavior in particular. And what they're showing pretty consistently is that when you look across the wide range of species, and when we're talking about songbirds, we're talking about literally thousands of species here. There are lots of birds where females are doing the same thing as males, but the hormones seem to be a little bit different. So again, in males in all these species, testosterone seems to be driving this behavior. Females consistently have lower circulating levels of testosterone. In canaries, you can get them singing every once in a while if you just leave them alone and you just record all day long and then take random samples. If you give them testosterone, now they're singing at a much greater rate. And so there's this idea, okay, maybe testosterone might be involved here. Maybe testosterone might drive and reshape patterns. But I want to show you the difference here. So here, I actually have an example from a study I did way back with my academic bestie, Farrah Madison at the University of Wisconsin. And so here we have the song of a castrated male. This male had been castrated for 3 weeks before we put him in the recording chamber. And this is what he sounds like without any testosterone, because again, if you're castrated, you're not producing hormones except for what's in the adrenal glands. And that's typically not enough to fully instantiate male typical behavior. So this is what that male sounds like. All right, that's cute, right? Yeah. It's very well put together, very structured. You can tell he's learned this thing over some time. Here's an adult female. We recorded this female for like 3 days before we got a song that was loud enough to record. So the females tend to sing lower amplitude songs, and because they're lower amplitude, they have to be conveniently facing the right direction in front of the microphone for you to actually catch it. And so this is what this female produced. Okay, now qualitatively speaking, do you think in your gut you could tell the difference between a male and a female song in this case? Yeah, so I got some head shaking, yeah. I know some of you are new to bioacoustics, but if you think about the signal, so here we have spectrograms of those songs. You read them like you do sheet music. The y-axis is frequency, so the higher the value, the higher the pitch. And the x-axis is time. And so you look at a staff is giving you frequency and as well as time, this time dimension. The color is giving you the loudness of that signal. And you can look kind of structurally, thinking about the physics of vocal production, you can see that the male is doing something that just looks different and orally sounds different than the female. If you give a castrated male testosterone or this intact adult female testosterone, this is what happens to their song. You get this output. So here's that same male about 2 weeks after he got some testosterone. So right in here, these are the sexy syllables, and that male just knew he was killing them with this little bit at the end. It's like I'm gonna lead in slow, but then I'm gonna really hit him at the end with a really nice little rendition. So here's the female, this is the same female about 3 weeks after testosterone. Okay, she sounds a little different. Now, again, when we're thinking about what is the meaning of an observation and how do we interpret this and then apply this meaning to some broader context, the traditional interpretation of this is that, okay, testosterone is masculinizing this female's output. So she's not only increasing the amplitude of her signal, but the structure of the song is qualitatively different. So this is not the same as this. This song versus this song, they are different, but there's some structural integrity that's similar between these two songs. So if I were to do a similarity score and actually isolate the individual notes and overlay and say, okay, physically speaking, the wave pattern of this sound versus this sound from these two different songs, I can see that there's high overlap. So the percent that's shared between these two songs from the castrated male is much higher than what you see from the intact female. So again, the idea is, all right, testosterone must be making this female like a male. It must be masculinizing her. That is one interpretation. But I want us to challenge that interpretation in particular, because again, thinking about where we started, we know that hormones are linked to reproduction. We know that they're linked to these social behaviors, these things that we do that facilitate, finding a mate, defending a territory, having progeny, raising those progeny. But the meaning of that, I want us to question the meaning a little bit deeper because there are some structural things that I think sometimes get lost in translation. In particular, when we think about, again, problematizing this, what does the female actually do in the absence of testosterone? So what I'm going to do next, I'm going to walk you through an example with European starlings. European starlings are across the whole of the US, basically. They were introduced, legend goes, in Central Park back when it was originally founded because of a Shakespeare company wanting to put on a play. And it's like, oh, we're going to release these starlings as to make it period appropriate in keeping with the text. And so those birds are released, and then now they just spread. They are an invasive species. So when we look at European starlings, the interesting thing with them is that much like other seasonal breeders, they go through this process where they're photostimulated, photorefractory, and then they go back to being photosensitive. Photosensitive means that they can then respond to long day lengths. This is a male's testes. So we look at a photostimulated male, you can see that there's rapid volume change, there's growth, and it's consistent across this phase. When they become photorefractory, so that means that they're no longer stimulated by long day lengths, they go back to basically a prepubescent state. So this is a testes from an adult male that is photorefractory. This adult male So I'm going to borrow a term that some of you know. That male is grown. Yes. But the testy is prepubescent because, again, this bird is photorefractory. And so when we're looking at what happens seasonally, there's this tight control of light and how light is influencing behavior. And in males, it's pretty clear that when they're photostimulated and up until the photorefractory, when the hormones are high and their gonads are active, that's when they sing. They're singing their butts off. They're singing as much as they can. And what we asked is if female starlings respond to testosterone across the season. And that's an important question because female starlings sing. Female starlings sing, but they don't sing during the breeding season. They sing during the non-breeding season. So right over here, when birds are photosensitive at the end of the breeding season, that's when they start singing. So they start singing around September, October, November, December, and then they wait for the long daylings to come back. And then once the long daylings start to come back around January, February, that's when they stop singing. So they stop right when or just before the males start. And so our question is if we have female birds who are technically in a non-breeding condition because they're photorefractory, but this is when males would have high testosterone still and they'd be singing. If we compare that to when they would naturally be singing, would testosterone be equally effective? And so the thinking is, okay, if it's like males, males would not be as responsive to exogenous testosterone because their T is already high. And once they've entered this photorefractory phase, it's like, okay, we need to shut the system down. We need to ignore the feedback loops. Let's just turn everything off slowly because we're about to go into the non-breeding season. And so they should not be sensitive. But they should be sensitive here if they're like males. So if they're like males, this is when you should start to see them actually starting to sing. Now, here's what we see. So here, this is brain output. We're looking at the volume of these different brain areas. So there are 4 regions that are critical players in the bird singing, the song control system. There's area X, L-man, HVC, and RA. In the birds that are given testosterone, they're in the light gray and black bars. And so the light gray bars, these are the birds that are in the post-breeding condition, so the photorefractory. The black bars are the photosensitive birds who are in pre-breeding condition. Both birds have been given T. Their brain radically changes. The volume of these regions, in some cases, triples. And when we compare that to the behavior, we get something a little funky. The behavior does not match what happens in the brain. The brain is suggesting that, okay, you have this volume change. The brain is radically different. That should facilitate some behavior change. But what we see is that only the birds that are pre-breeding begin to sing. Now, why would the birds that are pre-breeding begin to sing? Because everybody they had a brain change. Their brains are different in response to that. They're at male physiological levels of testosterone, but the birds who are in the post-breeding condition, again, the males are singing. Males in the post-breeding condition are still singing. So they're the males, starlings, March, April, they're still hanging out. They might be trying for one last clutch. But the females aren't responding in that same way. And so that's our first clue that maybe the females are doing something a little bit different with regards to testosterone. I did a different study, again, with my academic bestie, Farrah Madison at University of Wisconsin, and we looked at canaries in this case. And we treated male and female canaries simultaneously with 1 of 4 doses. We either gave them the control condition, a low dose, a medium dose, or a high dose. The low dose condition, where we're looking at serum concentration of hormones, this is not male physiological levels of testosterone. The medium dose is approaching male physiological levels, but it's not until you get to the high dose where you start to see male typical physiological levels of circulating testosterone. And so here we have this nice dose response curve where we're thinking about, okay, the effectiveness of our treatment. We can see this effective in both males and females. But when we look at the behavior, and this is the tricky bit, the sticky wicket, as I like to call it, the low dose here in these little dotted bars, on the y-axis, we have the proportion of time spent singing. On the x-axis, we have days. So we're looking at the first 7 days, so week 1 versus week 3. The low dose in the females are singing higher than the females with male physiological levels of testosterone. So again, just to jump back, the females in the high dose, so the females in the black bars here, during week 1, they have testosterone in the range of a male during breeding season. They are not singing. So the females with the high dose there in the solid black line, it's not until the 7th day of treatment where you really start to see them sing kind of as a group, but the low dose birds begin to respond kind of immediately. One thing I should say is that all of these birds are photosensitive. So they're being held on short days. So you can think of it as a pre-breeding condition, right before the breeding season actually starts. The males at each dose point are responding, but the efficiency of the response also seems to be better with the low dose and the medium condition. So there's some significant differences here in this first few days, but then all of that kind of washes out by the end of the first week. But then when we get to week 3 with the males, the more T seems to be the better. So the testosterone levels that is most closer to the male typical physiological levels yields the best result long term. Short term, there's some funkiness here, but again, you think about treatment and dose responses. Short-term treatment sometimes yields a little different result than long-term treatment. Long-term treatment shows what you would expect from males, that more T in the physiological range yields a more voluminous output in terms of singing. The females are all over the map. Every dose response yields a similar result. It's not just the high T group, it's the medium group and the low group long-term. And so the low group in particular, when we think about like what that means, I did the study with an undergrad this summer, and we're currently in the process of writing this up. So we looked at hormone base because one of the problems with a lot of the songbird data is that we don't have a lot of solid data around what females are doing. But conveniently, in the hormone-based database, which is a publicly available data set, they do have included where studies have measured female concentrations of testosterone. And so what we did is we divided all of the species that are available on that data set into 2 groups. Either song is present or song is absent. So in the species where song is present, those are represented by the gray bars. And species where the song is absent, they're represented by the black bars. The hormone-based database does not have every songbird. There were about 61 total species that fit within these 2 categories. And so with the birds that were present, that's about 21 species. The other 40 species are in the absent group. But when you look at the mean level of testosterone concentration, so here we're looking at just circulating T in females. So wild-caught females, that's what the bulk of these were. There were a couple of lab studies. In the species where females sing, testosterone tends to be higher, but not in the male physiological range. So you compare females that sing versus females that don't sing. Females that sing have a higher level of T, but it's not at male-like concentrations. Here we're looking at the coefficient of variation. So we're looking at across studies, the stability of these measures, and there's no difference in terms of the coefficient of variation for each of these species that are represented in this data set. So we can be pretty secure in our observation here that it's not due to some sort of distribution error. Now, interestingly though, when we look at basal cortisol, we see that birds where females don't sing, they have higher circulating corticosterone versus birds where the females do sing. And this kind of fits into a larger hypothesis that's called the dual hormone hypothesis that suggests for particular behaviors, there's this linking between testosterone and corticosterone where, and usually this is talked about in terms of males. So for certain male behaviors to occur, they got to be relaxed. They got to be chill. They have to have low cortisol. And then you can have higher levels of testosterone that facilitate these behaviors. So when we're thinking about things like status seeking, dominance and aggression, in high status males that are dominant and aggressive, they tend to have lower cortisol, lower corticosterone in animal models, and higher testosterone. We're seeing a similar pattern in the birds, but just to backtrack, they're at a much lower physiological level. And again, think about why all this is important. So again, when we think about hormones, females use testosterone as well. So when we think about sexual behavior and what might be controlling sexual behavior, there's a role for testosterone in female sexual behavior that persistently goes unaddressed. So in thinking about sexual health, one of the classes I teach is human sexuality. I spend a lot of time talking about perimenopause and postmenopause, and thinking in particular about hormone therapy related to treating those conditions. And the problem with a lot of things that happen in the States is that including androgens as a part of HRT is not standard care for most folks in the States. In the EU, they do include androgens as a part of the standard panel of hormones for hormone replacement therapy, but not so much here in the States. And the question I have is why, if testosterone is involved in female sexual behavior, if female, and again, thinking about the level, it's not a lot. It's just a little bit that you need to get these pattern changes. So to instantiate, to get the behavior going, you just need a touch. So again, thinking about that, a part of that I think has to deal with how we think about hormones and how we talk about hormones. Just to kind of wrap up here, there's a study by Daphne Joel that was published in 2015 that I often use in teaching. Again, going back to this concept of when we talk about brains and behavior, we talk about things as though they are dimorphic, when typically there are differences. So male behavior here, female behavior here, most of the time there's overlap. And that overlap, because we're talking about averages, we tend to ignore. And so again, this is thinking about like philosophically and rhetorically, how do we communicate this science? This is the sort of the main thrust, the main argument that I'm making here, because when we think about how we educate folks, the discourse around neuroscience, the discourse around hormones, the discourse around sex and sexuality, there's a need for a more inclusive model because the data is telling us that there's a need for a more inclusive model, right? Similarly, speaking in terms of thinking about the application of this and how do we correct our thinking around this, this also has direct policy considerations. So this is a brief blurb from the abstract from a paper I published not too long ago, thinking critically about how policy is influenced by our misinterpretation of science. When we're thinking about how do we teach and talk about this stuff, that actually does have a social impact. So beyond what the data is saying, there's a real consequence when we don't give nuance to what we're actually observing. And again, that's important because when we think about how we talk about sex, so the discourse around sex hormones, reproductive biology, and the organization of brains, all of this is going to be contributing to how we treat gender diverse populations, how we think about, even in basic science, why is it controversial to have a female model of behavior when for decades and centuries there were only male models of behavior? Yes, there were initiatives to make it standard to include males and females in studies, but there's still a pushback that some folks get when they say, I only want to study female animals. And thinking about the why behind that, that's a critical evaluation. But then thinking a bit further, because we use science to then help ourselves and think about the human condition, with regards to reproductive health, sex education, and understanding peri- postmenopause, which thankfully we're finally starting to discuss as a population, we also need to think critically about how we're talking about hormones. And how do we, as folks who can influence policymakers, how do we get them to start thinking about that as well? And so that is my cue to give the thank yous to all of my collaborators and contributors, as well as my lovely students featured in this talk, Evan and Callie. And of course, the people with the money, because I don't have very much of it. Yes. All right. So, any questions?
I'm gonna stand next to you so people on Zoom can hear. Are there questions from Zoom?
I don't see right now.
Okay,
But it might help you repeat any question that.
Yeah. so if something comes, just repeat.
Yeah. Yes.
See this to animals with more complex.
Yeah. So the interesting thing is with mammals, I would actually argue that birds also have very complex social structures. So take, for example, zebra finches. Zebra finches in the wild live in colonies of hundreds of birds. So you can think of it like a small college campus, right? Everybody knows everybody, but not everybody hangs out with everybody. So similarly with zebra finches, they have smaller social networks, so clusters of like 9 to 10 birds that are sort of mini units. And so within the larger superstructure of the colony, you have these subunits that then communicate reliably with one another. And so thinking about the complexity of how do you maintain those specific kinds of social bonds, I would argue that there's actually a lot more complexity in animal social systems than we typically think of. But sorry, I don't know if that addresses your question. Yeah. Yeah. Yes.
Have there been any studies like using estradiol birds?
Yeah. So right on. So have there been any studies introducing estradiol similar to the studies with testosterone? And the answer is yes. So Elizabeth Atkins Regan did a lot of this work in the early, mid 1990s with her grad students. And so what she did is she looked at female zebra finches because zebra finches are actually dimorphic. They are one of the rare cases where you can say this is a true dimorphism. Males do this, females do this. If you look in the brains of female zebra finches, it is nearly impossible to even find area X, which is a critical part of the basal ganglia. If you give a female zebra finch estradiol within the first 2 weeks of life, then you masculinize her brain. And if later on you give her testosterone, then magically this adult bird can sing. And so there's a specific role for estrogens in masculinizing during the perinatal period. And so once you're out of that sort of 2 week window, then estradiol isn't going to be able to actually do that job of masculinizing. And so there are some studies that sort of show, again, and sort of problematizing how we talk about hormones. Again, there are no male or female hormones. Hormones are just doing different jobs at different time points. Right on. Thank you for that question. Here and then here. Yeah, in part that was the original idea because when I first got into Songbird, the question was, how did I get into using songbirds as a model species? And a part of the question was about, okay, thinking about sexual different behaviors. And that originally was a part of it because in reading about it before I even applied to go to Hopkins, because I actually stalked my PhD advisor for like a year, and we actually communicated over email, old-fashioned letter writing over the course of a year, where we would talk about science and the data, and he'd have me read papers, and I'd come back with a question. And one of the things that I kept seeing was like everyone's talking about sex differences, and there seems to be this clear difference between male and female birds, and that is interesting. I actually want to probe that and see if it's true. And so that kind of drove me into the bird world. Yeah. And you had a question. Yeah.
So is there a dimorphism at all in, say, our canary friends in the strings?
In the
vocal apparatus? Is it maybe more developed in the adult male?
Right.
Is there a developmental pathway that
Right.
can follow?
So the question is, if you look at the syrinx in canaries, are there going to be sex differences that you can observe? And the answer is yes. So the syrinx is different in male and female canaries. So the male canary, because the syrinx is actually very sensitive to T, there are androgen receptors in those muscles. They are much larger in the male bird. And so one of the thinking is that if you treat a female bird with exogenous testosterone, that might be actually changing the structure of the syrinx itself to facilitate, actually producing sexy syllables. But then that also begs the question, if the females don't need the sexy syllables for singing, so how are they using song? What's the function of it? Then would they even need that well-developed syrinx? So again, thinking about what's the function, because they do sing, they just don't sing very much and they don't sing very loud. And we don't really know why. Yeah.
I'll let you read it just because it's got a couple words.
Gotcha. The question is, does the pineal mediate the effect of light in short days / photostimulation on the hypothalamic pituitary gonadal feedback system? And the pineal is involved, yes. So we have deep brain photoreceptors that actually respond to light. So the skull of the birds are not translucent, but the light is able to pass through. And so you can actually get light reaching out into deep brain areas. And so you have these opsins that react to those daylings, and you can see activity in the pineal responsive to the photostimulation. The second question is, the most prominent brain effect of testosterone suppression treatment in men with prostate cancer, measured by psychometric testing, is a negative influence on visual spatial functioning, among others. Is there any evidence among androgens regulating visual spatial function in birds? This capacity must be important among bird species to be able to recognize each other. Right on. That is a good question. So in birds, there isn't a ton linking hormones directly to sort of visual spatial efficiency. I'm struggling to think of any direct sources, but that is actually a good observation. So we think about hormones and systems. One of the things that question makes me think of is a good colleague in grad school was also studying sex differences, but they were actually studying them in humans, and they were looking at differences in visual spatial abilities. And her argument, so this was from Amy Shelton's group and her graduate student at the time, who's my friend, also named Amy, weirdly. Amy S. too, weird. But Amy's big question was, is there actually a functional difference in the visual spatial abilities of men and women, or is there a strategy difference that's related to how the brain actually processes visual information? And she actually had a good bit of data that strongly suggests that it actually is a strategy difference. So how we mentally rotate images and navigate visual spatially. And so it's not necessarily a sex difference, more so a strategy difference. And thinking about how hormones might play into that, it might be that the hormonal profile facilitates different strategy choices that then lead to different patterns in terms of outcomes. Right.
Don't go anywhere. Thank you so much. That was really informative. I'm at the mic too. Everyone, please. Thank you, Dr. Rouse, for being here. You're great talk. Good job. And we're around for a few more minutes. If you have an individual question, anybody?